The overall goal of this procedure is to dissect and culture mouse embryonic pancreatic buds ex vivo, allowing direct visualization of the developing pancreas. This is accomplished by first dissecting embryonic day 11.5 or 12.5 mouse embryos and removing the upper body and tail region to isolate the mid-body of the embryo. The second step is to expose the duodenum region and the stomach for location, and then dissection of the dorsal pancreatic bud. Next, the bud is transferred into the microwell of a MacTech dish and cultured for up to 1 week. Finally, the explants can be analyzed for live cell imaging, or the explants can be fixed, whole mount stained, and analyzed by immunofluorescence imaging. Ultimately, pancreatic cell differentiation, cytoskeleton organization and cell polarity, and branching morphogenesis can be evaluated.
Ex vivo culturing of pancreas explants can help to answer key questions in pancreas organogenesis, such as branching morphogenesis, how does it take place in the developing pancreas, and how this event is connected to growth and differentiation.
On the day before the experiment, coat the 20 mm diameter glass bottom microwells of 35 mm MacTech Petri dishes with about 150 microliters of fibronectin, covering the whole glass surface. Then, place the dishes in a 4 degrees Celsius refrigerator